Okay, uh, let's get started. Nobody has joined, but I have to start the session. Okay, wait a minute or two. 1 second. Okay, let's get started. Oh, sorry. Yeah, let's get started. So, welcome to week 5 of Advances in Omics. Uh, in the last weeks, we have discussed different types of sequencing technologies and how we can use them to find to predict evolutionary constraints that has gone through the years and so on. So today we will talk about mainly we focused mostly on genomics in the previous classes. Today we'll focus a little bit more on the other types of omics, which are transcriptomics and proteomics. So yeah, let's get started. So yeah, this we talked about in earlier classes about how conservation of genomic sequences can actually tell you about several of evolutionary caveats. So if we think it from a protein perspective or an amino acid perspective, that is also kind of true. But yeah, here we are talking about genomic sequences again. So you can have homologs, orthologs, or paralogs. So basically, sequences from which certain mutations have gotten rise to and then diverged to form different species or different types of so for example, humans and chimpanzee will have uh, a lot of same sequences in genes, but certain loci might be changed, actually tells us about which actually tells us about the uh, tells us about how evolution wise yeah, we have evolved from chimpanzees or monkeys. So the idea is to identify these related genetic sequences. You annotate them if these are CPG-rich islands, or if these are exonic regions of the genome, or these are just repetitive elements which actually are some sort of variants in the genome. So we annotate these reference sequences, and then we do alignment. So you can have local alignment or global alignment or multiple sequence alignment across different species of the same gene or the same region of the genome, and then you identify different conserved sequences. So when you do multiple sequence alignment, some region of the genome will be highly conserved, they are not changed, but some have changed a little bit due to evolution and adaptation to the environment, and that is how different species get different characteristics, right? Then we kind of visualize the sequences, and we try to pinpoint which regions of the genome are conserved and which are not conserved, right? So for example, this picture is taken from Wikipedia. So these are like histone sequences, these are protein sequences, these are not genomic sequences. As I said, genomic sequences and protein sequences, you can like for understanding adaptation and evolution, we can use both of these types of sequences to see how we have diverged from our ancestors, right? So here are like five organisms: humans, chimps, mouse, rat, and cow, and these are their sequence of histone H1 protein. So you see there are some certain regions of this protein which are highly conserved, and there are certain regions where we have differences. So for example, in the first one, in this particular position of the amino acid, humans and chimps have same, mouse, rat, and cow have different sequences. Similar in this case of in the case of this particular sequence. So these are uh, this is how, to get to this level, we have to do multiple sequence alignment in a global or a local reference, so that we can find this set of regions which are conserved. So some are semi-conserved, some are not conserved, at non-conservative. So basically this is almost conserved, but in one species there's a change in the amino acid, right? So yeah, so the idea is that sequence conservation implies function. So basically, what it means is, if a sequence is conserved highly, it means that the region of the genome, or the region of the protein, or the set of amino acids, or the set of nucleotides actually code for something very important, which is fundamental to the downstream protein, or downstream processes. And then we have slight modifications depending on the adaptation to the environment, and so on, evolution, and so on. So this is again an example of like mouse. I don't know if you can see this. So mouse, chimp, human, dog, and rat. So you have sequences which are highly conserved. So this red line tells you that these are very highly conserved regions, and there are regions which are not highly conserved. So you can do pairwise comparison, say for example between human and mouse, or human and chimps, or you can do multiple sequence alignment, taking 5-10 species together and then seeing what is the conservation. To do that, we are as we talked about, alignment. You have to do sequence alignment, and you have to find important regions which are determining functions of the genes or the functions of the proteins, and to basically understand what are the evolutionary forces that actually drive this particular set of conservation or mutation, right? So yeah, going forward from that, we'll, as I talked about earlier, that we'll be focusing today more on the other omics, which are transcriptomics and proteomics. We have talked a little bit about genomics across the first 4 classes, but this one will be mostly focusing on transcriptomics and proteomics. Now, the word transcriptomics comes from the word transcriptome, which is basically a complete set of transcripts in a cell, or a population of cells, or a specific developmental stage, or a physiological condition. So you define a condition, so that may be a disease condition, that may be some sort of experimental condition that you have generated, and at that condition you check what is the transcript levels of the cell. So transcripts of different genes getting expressed in the cell, what are the levels? Are there fluctuations? Is it the same as a control, or are there deviations which can actually tell you the underlying condition of the cell, be it disease, be it some sort of other physiological conditions, right? So in general, this can be summarized as the total RNA content of the cell, but uh, be a little careful. When we talk about transcriptomics, we mostly talk about mRNA, because that is what gene expression is. So the conversion of the genomic sequence of a gene, of the exons for eukaryotes, to the mRNA. Uh, so this step is known as gene expression, right? So we mostly look into gene expression through transcriptomics, but also you can do sequencing of other types of RNAs as well. So transfer RNAs, rRNAs, but generally we do for mRNAs. The maximum papers that you'll see, the maximum work that has been done and in transcriptomics is focusing on mRNAs, and it's maybe a small amount of non-coding and small RNAs, right? It is often focused around the level of, or the changes in levels of transcriptional activity at each locus. So we look for gene expression levels, transcript usage within a locus, allele specific expression, preferential expression of a parent allele, EQTL, which are expression quantitative trait loci. Sorry, I think there's some network issue. Okay, I hope you can hear me. Please let me know in the chat if I'm not audible. Okay. Yeah. So again, what are so these are very important things: EQTLs and allele specific expression and so on. So the idea is that you have the whole gene. Oh, okay. Thanks. You have the whole gene, and then you have, say for example, you have one position mutated in the gene, right? So maybe a single nucleotide variant that has that is some synonymous in nature, and one position has been mutated in the gene. Now while doing the genome sequencing, you have identified that particular mutant in the gene, but you do not know what that mutant actually is doing inside the cell. What is the contribution of that mutant to the genome, to the downstream processes of the cell. So for that, what you do is do RNA sequencing of that particular of individuals or cells that contain that particular specific type of variant, and then you do RNA sequencing of a controlled set of cells or individuals which do not have that variant, and then you kind of compare between the two in terms of mRNA levels or transcriptomic levels. Are there any differences because of this particular variant? So if a particular genomic variant actually changes the expression of a gene downstream, we call that EQTL. So that becomes very important part of the discovery, because it is directly impacting the expression change in the cell downstream, right? Also, we use different types of transcriptomic technologies to find de novo, new transcripts, and basically do de novo transcript assembly as well. Okay, so just to give a primer about mRNA processing, like what how does mRNA look like? So this is the DNA, where you have exonic regions, intronic regions, and there is a transcription termination site. Upstream is basically a promoter, and a transcription start site, right? So what happens is the DNA polymerase comes and binds to the promoter and makes the mRNA out of the DNA present here, and then this is what is the primary transcript, in the 5' end of which you have a cap, and the 3' end you have a polytail at the end. So once the transcription is done, then what you do is known as splicing. You remove this set of introns from the middle, and then you join the exons. So this whole process is called as transcription. I hope you know this. So from the DNA, you go to the mRNA, which is the spliced mRNA, after removing all the intronic regions. The important thing is that on the 5' end of this mRNA, you have a cap, and on the 3' end you have a poly A tail, a set of long A tails at the end, right? So how do you kind of measure this transcript levels in the gene? So initially, again, I think it is written. It was in 1991 that this particular type of not technology, this particular type of method, I'd say, was used to quantify the amount of transcripts in a cell, right? So they sequenced mRNA brain sequences, and they use this something called as expressed sequence tag. What it is, it's basically if you take this particular mRNA, and then you do reverse transcription on this, and then you find a complementary set of nucleotides to this particular mRNA, and then you put that complementary set of nucleotides in a flow cell or in an array, and then see if your particular cell has that particular mRNA or not. So in your case, you'll have this particular sequence, and you'll check if this particular sequence is coming and binding to the to your sequence, right? So if that is happening, that means that that particular transcript is getting expressed in your cell. If that the hybridization is not happening, you do not have. So again, one thing to notice that ESTs are basically DNA sequences. These are not RNA sequences. So mostly when we talk about transcriptomics, we are talking about RNA sequences, but in most cases this RNA sequence is not directly sequenced, right? So you have the RNA. What we generally do is we use a reverse transcription enzyme and convert this RNA to a complementary DNA, and then we sequence the DNA, right? and then we like convert the information back to what RNA sequence it was. So here also it is a similar case. You have a complementary DNA, right? Complementary DNA uh, to the RNA, which is actually getting converted to cDNA by reverse transcription enzyme. So here we are talking about, although we are talking about RNA sequences, but we do the sequencing mainly through DNA, because the DNA is easier to sequence, and like we are more kind of advanced in terms of DNA sequencing than in terms of direct RNA sequencing. So in general, we hybridize it with a set of complementary set of DNA, and then we sequence this. So this is a simple idea that was initially used, ESTs, which are expressed sequence tags. This is used till now, till date as well. So but the idea is that you have a, a, a limit on the length of the RNA that can be sequenced. So it is around 100 to 800 base pairs is the length of the express sequence tag DNA. So the amount of RNA that you can the length of the RNA that you can sequence is probably around this. But again, the idea is that you know what is the sequence length and the whole sequence of the RNA is. In today's date, if you want to do an EST, you know that a particular mRNA of a particular gene will be this much in length, and this is the sequence. You want to find if that particular mRNA is getting expressed in your cell or not. So all you need to do is get this particular signal, this hybridization, and in downstream a fluorescent signal to tell you that this particular mRNA is in the cell and it is getting expressed. And if you have more signal intensity, that it is getting expressed more. If you have less intensity, it is getting expressed less. So that is the whole concept of EST, right? Now moving on to microarray technologies, which are a little bit more advanced than ESTs. So here also the idea is similar, that you have so let us go by this. So you have a sample, and then you kind of purify the mRNA and the protein and the DNA from the sample, and you take the mRNA. Now after taking the mRNA, you use the reverse transcription enzyme on this, do the reverse transcription from the mRNA, create the complementary DNA. This cDNA is not circular DNA or something like that. This is complementary DNA to your mRNA, right? Now you label this cDNA with some dyes or some fluorescent signals and so on, and then what you do is a hybridization step. So you have an array. In the array you have these different signaling, not signaling. So these are basically sequences of DNA that are actually complementary to the sequence of DNA that you have generated here. So if this DNA particularly goes and binds to some of your preset DNA sequences in this particular chip, it'll hybridize and it will release the fluorescent molecule, and you can capture that that signal, that this hybridization has taken place. And then again you wash out the excess, and then you capture the signal, and basically you actually know what a particular mRNA is present in your cell or not. The major idea here is the key assumption here is basically that the fluorescence is proportional to the mRNA abundance. So how do you say that a particular gene is getting expressed more or less? The more or less concept comes from the signal intensity, right? So if you have multiple of these transcripts in the cell, and if you put them on the chip and the signal intensity is very high, that means your gene is getting expressed in higher numbers. And if it is on the lower side, then means your gene expression is lower, right? So you have a control sample and you compare it with the control sample. Is it higher or is it lower? So yeah, the mechanism of detection is kind of similar to DNA sequencing, that you have fluorescent labeled mRNAs which are converted to cDNAs, and then it goes and binds to the target sequences, in this case in the chip, in terms of hybridization, and then you detect the fluorescence with some sort of scanner or some camera, right? So, and the amount of fluorescence is actually proportional to the mRNA present. Now, we talked about ESTs, then we talked about microarrays. Now, this next step of the technology advancement brought us to RNA sequencing, right? So what are the fundamental differences? Why do you want to do RNA sequencing compared to microarray? Because microarray looks simple enough, right? The difference is that in microarray, probes are designed with a prior knowledge of the target sequence, because you know a particular sequence of RNA and you just want to find out if that RNA is getting expressed in the cell or not. That is all you want to do. So you need to have a prior knowledge what the length of, not the length, at least what is the sequence uh, of a major portion of that mRNA, so that you can create your target, and after creating the target, your basically mRNA from the cell can come and hybridize to this particular target and give you the signal. So to do this hybridization, you all actually have to know what the sequence of the DNA is that you are trying to sequence of the RNA is that you are trying to find. On the other hand, for RNA sequencing, it is basically we will it will contain specific sequence information. So in microarray you do not have specific sequence information, right? So you only know a particular length of the sequence, but after that you do not need to know, and you like kind of um, do not know even what the sequence is. But in terms of RNA sequencing, since you are sequencing the whole um, length of the RNA, you have to know what is the you will get to know what is the sequence of each of the position of the RNAs, right? So it contains specific sequence information. We count the amplified fragments from source material. We can detect and quantify novel or unknown sequences. We can do de novo assembly of a transcriptome. We can find novel transcripts and find where the transcripts actually came from. And also we can detect alternate isoforms of the DNA of the mRNA as well. So here I was talking about the splicing part, right? This splicing is not constant for every gene product, right? So you'll have different alternative splicing. In some cases, both the exons will be removed, introns will be removed. In some cases, one intron will be removed, the other will be there, or like so on and so forth. Not the intron will be there, the intron will be removed. How many introns are getting removed and what is the length of the mRNA, that can vary depending on the downstream requirement of the cell, right? So you'll have multiple isoforms of the same gene, right? That you cannot detect with microarray until and unless you have the prior sequence known. And for that you need to know the whole sequence, because two isoforms will have very similar sequence similarity, right? So that can be easily done using RNA sequencing as well. Excuse me. Okay, so coming to RNA sequencing, you have a long mRNA with a poly A tail at the end. You fragment this mRNA and do reverse transcription to find the complementary cDNA, right? Then you uh, cut down these smaller fragments, and then you put ESTs, expression sequence tags, with adapters, and then you do like basically a DNA sequencing on this to find what is the sequence of the complementary DNA. If you have the sequence of the complementary DNA, you can get back what the sequence of your RNA, mRNA for that complementary DNA will be. So, so long RNAs are first converted into a library of cDNA fragments, either through RNA fragmentation or DNA fragmentation. Sequencing adapters are subsequently added to each of the cDNA element, and a short sequence is obtained from each cDNA using high throughput technology. The resulting sequence reads are aligned with the reference genome or the transcriptome and are classified as three types. They are either exonic reads, which are actually coming from the part of the genome which is actually coding for the protein. There can be junction reads, which are like between so exon, and in between there is an intron, and you have a read which kind of is spanning both. So those are junction reads, and then you have the poly N reads, which are actually which is a poly A tail of the mRNA, right? So you will have these three types of reads, and these are used to generate a base resolution expression profile for each gene. And this is an example of East ORF with one intron getting shown. So these are this is your expression level on the y-axis, and this is the nucleotide position. The expression level again is measured basis on the basis of how many counts of RNA is how many counts of that particular location is getting detected. Okay, so this currently this what we are talking about is mostly short read technologies, but now we have long reads as well, as we talked about in earlier classes. Oxford nanopore technologies, PacBio technologies, and so on. We have long read sequencing nowadays, so we can do RNA sequencing with long reads as well. So uh, uh, the short and long read cDNA methods share many of the same steps in the protocol, but all methods require an adapter ligation steps and are all affected by sample quality and computational issues upstream and downstream of library preparation. So three main sequencing technologies here shown. So this is your, if you remember, this is your Illumina sequencing. You have bridge amplification and hybridization for each cycle, and in each cycle you capture the base that is getting read at that position. PacBio and ONT, you remember we check for ion current changes due to the passing of the nucleotide at the pores and so on, right? So if you do a comparison of short, long read, and direct RNA sequencing analysis, so basically direct RNA sequencing is not converting it into this complementary DNA and doing the sequencing of RNA. That is still possible, but more widely used is basically complementary DNA sequencing. So what can happen is basically if you have long reads, you can uh, find you can span the junctions between exons and introns, and you can find different isoforms of the mRNAs much easier compared to short read. So over 90% of human genes are alternatively spliced to form two or more distinct expressed isoforms, so in this case transcript X and Y. The complexity of information captured increases from short read DNA sequencing, where isoform detection can be compromised by reads that cannot be mapped unambiguously, to long reads that directly sequence isoforms. In short read cDNA sequencing, a significant portion of reads map ambiguously when an exon is shared between isoforms. Read span exon-exon junctions can be used to improve the isoform analysis, but can also be mapped ambiguously when a junction is shared between these isoforms. So due to this complicates the analysis and interpretation of the result. Long read cDNA technology can come in here, and it can generate the full length of the isoform read that remove or substantially reduce these artifacts and improve differential isoform expression analysis. So for isoform expression analysis, this is very useful. But other than that, the technology is kind of same. It's just the length of the read is higher. Long cDNA, so it can reduce these artifacts and improve the detection of these isoforms. However, these methods rely on cDNA conversion, which removes information about RNA-based modifications and can only make crude estimates of polyadenylation tail length. Direct RNA sequencing enables full-length isoform analysis based modification and poly tail length estimation. So again, if you remember, for DNA sequencing, we also had DNA modifications, which are your epigenetic modifications, histone markers, your acetylation and methylation of the DNA. So that information we can capture along with the sequence of the DNA using long read technologies like ONT. But in case of in case of RNA sequencing, the issue here is basically the RNA modifications that are present on the RNA, because again, post transcription there will be a lot of modifications, a lot of compounds can be added to the RNA to make it stable and so on. So that information will eventually get lost because you are converting the RNA back to a complementary DNA and you're removing all these modifications and before sequencing. So that is one of the major caveats of complementary DNA sequencing, that you cannot get a full picture of how the RNA is actually in a chemical state. So if it is modified, if it is not modified, if and these modifications can tell you if it is active or inactive or so on, right? So that information is missed by complementary DNA sequencing, but again, if you do direct RNA sequencing, you can get hold of that information as well. So yeah, coming to direct RNA sequencing, so this is an example of direct RNA sequencing done on MinION. So MinION is basically a machine from Oxford Nanopore again. So they have done a sequencing of 16S rRNA. So you have a, a 16S rRNA that you are wanting to sequence. So you add a sequencing adapter bearing RNA motor protein and hybridize and ligate it to the 3' overhang of the 16S RNA, 16S rRNA, and then the sample is loaded into this particular flow cell for sequencing. So representative ionic current trace during translocation of this 16S rRNA strand from E. coli through the nanopore. So basically, again, the idea is similar, that if you have if you pass your sequence through a pore and it changes the current, amount of current is changes and how much it changes by, will tell you what particular base is at a particular location, right? So yeah, following the capture of 3' end of a adapted 16S rRNA, the ionic current transitions from open channels to a series of discrete segments characteristic of adapters. This is followed by ionic current segments corresponding to base by base translocation of the 16S rRNA, and it can be and the trace is basically this trace is representative of thousands of reads collected for individual 16S rRNA strands from E. coli. Sorry. Yeah. So yeah, the idea is that you do this. Firstly, one thing you have to notice that RNA is always single stranded, so you have to make it double stranded. So for this, they do a hybridization step and they pass it through a nanopore. So the idea is similar, that you, when it passes through the nanopore, the change in ionic currents will tell you what particular base it is, and also it will tell you what modifications on the RNA is present or not, right? So that is a very good idea to do direct RNA sequencing, but it is much easier to do DNA sequencing because DNA is much more stable chemically and to heat. So it is very difficult to do RNA sequencing directly than to convert it back to DNA and do the sequencing. So for most cases, you'll see complementary DNA sequencing instead of direct RNA sequencing, but it is also possible here. Okay, now what we have been talking about is sequencing of a particular sample, right? Now what a sample is basically a collection of cells of an individual or of a cell line or so on. Now if, say for example, you want to check the expression of a particular gene in the liver, right? So you take a liver tissue, you get a sample from the liver tissue, and then you do RNA sequencing on that, and then you have a control and you have a disease state, and you have similar amounts of the samples taken, and then you do a comparison head to head if some genes is expressed lower or some genes is expressed higher. But the idea is that not all cells, biologically speaking, not all cells will have the same amount of expression, right? The expression is particularly in such big tissues, let's say for example liver or brain or say immune system, your blood cells. So the expression of particular genes will be cell types dependent. What the cell type of the tissue is, the gene will be expressed in such a manner. So for example, if you are doing some transcriptomic analysis of the brain, you will have neurons, you will have microglia, you will have interneurons, you will have astrocytes, and so on, right? So these cell types will not like express all the genes among themselves in a similar manner, right? Because the cell types have specific function, and depending on the function, the gene will be expressed. But what you are calculating for these RNA sequencing experiments through these kind of experiments is basically an average of all the cells that you have taken in a sample. So if you have 1 million cells in the sample, it can be possible. If you have a million cells in the sample, the expression values that you're getting is basically an average of all these million cells, and it does not tell you the heterogeneity of expression among different cell types of that particular tissue, right? So there the concept of single cell transcriptomics come. Here the idea is that you take RNAs from a single cell. You can have multiple such cells. You can have like a million cells, and right now we are capable of doing million cell single cell RNA sequencing. But the idea is that you take the RNA expression or the gene expression of each cell separately, because each of the cell type will have a specific type of expression, and you want to capture that heterogeneity. So again, why do we need this? So because tissues are not uniform, they contain diverse cell types and states. Single cell transcriptomics allows for fine-grained analysis of these differences, revealing the unique gene expression patterns that define various cell populations within a complex sample. The average behavior measured in millions of cells does not necessarily reflect the behavior in individual cells. So this is the important part, that what you are getting from normal RNA or bulk RNA sequencing is basically an average of all the cell types present in your sample. But that does not tell you, give you a clear picture of inside this particular cell types is the expression more or less, right? It gives you an average. So that is, a lot of information is getting lost there. So also single cell is crucial for finding and characterizing rare cell populations that might be missed in bulk analysis, but could play significant roles in disease progression or development. And we can also use mapping mapping for cell lineages and trajectories. So for example, you start with a stem cell, and then you kind of grow that cell into a particular type of a tissue. So this whole trajectory for different so say for example this takes 90 days, right? So from starting from a stem cell to a particular type of cell, this takes, say for example, 90 days, and you do single cell RNA sequencing at like say 4 stages, right? And after this you can have a trajectory of how this particular stem cell actually went on to become a particular cell type. What changes in gene expression happened, which genes got expressed more, which genes got expressed less, and so on. So studying these individual cells over time during development, we can reconstruct cell lineages and understand complex pathways that lead to different cell types. The cell differentiation is a very important part of single cell transcriptomics, transcriptomics applications, right? Yeah, so this is, again, I'll not go into much details of this. So the idea is that you'll have expression information of all the genes of one particular cell, and you'll have such information for, say for example, if you're sequencing 10,000 cells, so for 10,000 cells you generate this in FASTQ files, or you have count matrix, and then you kind of do normalization, and you see which genes are getting more expressed, less expressed. Again, so since we are quantifying the length of the the number of the transcripts that is present in the cell, the number of transcripts is also going to be proportional to the length of the gene as well. So we generally normalize using gene length and so on. So we do a lot of normalization to actually find out it is actually getting overexpressed or underexpressed, or is it same? It's just because the length of the gene is more, we are getting more expression. So for that, so after that we do downstream analysis, we do clustering of these cells based on these gene expression values, and the idea is that similar cell types will cluster together and different cell types will cluster in a different manner, right? So yeah, this is the idea. Now, how do you identify different cell types? So we do something. So imagine what your data looks like, right? So for example, you have done single cell RNA sequencing on say 20,000 cells. So you have, if you think of a text file, a table, right? So all your columns are basically your sample IDs. So you have 10,000 of these columns, and on the row names, or the first column will contain all your genes, right? So say for example we have around 18,000 genes say for example, right? So you have 18,000 cross 10,000 matrix, which is your data set. So these 18,000 genes, what is their transcript information, transcript number of transcript or normalized transcript value, transcript count for that particular sample? You have that for 10,000 samples for each of these genes, right? So this is your data set, which is 10,000 by 10,000. Now what you want to do is do a dimensional reduction. So you want to capture the variation of this 10,000 cross 10,000 in 10,000 cross 2. So you have 10,000 vectors explaining the diversity or the variation in the whole data. Now you want to collapse 10,000 columns into 2 columns. So that is what dimensional reduction is. So we have different techniques: principal component analysis, UMAP, TSNE, and so on. So this is what a TSNE looks like, right? So you cluster, so you condense the information into 2 vectors, and then the 2 vectors are TSNE1 and TSNE2, and then you kind of plot a scatter plot, right? So the idea is that similar expressed cells will cluster together, and if they cluster together, mostly they'll be of the same cell type. So for example, this particular cluster has clustered together. So this is some sort of a H9 cell. So for example, the green one, this is some sort of a muscle cell. The darker, this is also green, probably the darker green ones are your liver cells, and so on, right? So we do different types of clustering techniques: PSCA, TSNE, UMAP, and so on. And so the idea is that after clustering, you'll have all these clusters. Now how do you identify that particular cluster is actually a particular cell types? So for certain cell types, certain genes will be overexpressed or underexpressed. Say for example, some genes will act as markers for particular cell types. So for example, say B cells or some immune cells, CD36 will be a marker of the immune cell, because CD36 would be highly expressed in these cells compared to, say for example, some neurons. So you can differentiate between these two on the basis of clustering itself. Also you can color these clusters based on this expression of particular cell types, and you can identify, so okay, so this set of cells or this cluster of cells are expressing this particular gene more or this particular gene less. So that can act as a marker for identifying that particular cell type. So that is how single cell RNA sequencing data analysis is performed. Right. Now also you can plot trajectories of different cell types across developmental stages, as I talked about, and you can find a point where it is actually diverging. So this is, say for example, all the cells started from here and it went up to here, and after that it is diverging. So you can find the point at which it is diverging, what is the gene expression at that point, why biologically this might be happening. So this is actually a very powerful tool to understand the underlying biology of why and how this is happening, right? Okay, now we come to something called RiboSeq. So it is very similar to RNA sequencing, but it is the principle of RiboSeq. Why we need RiboSeq if we have RNA sequencing and single cell RNA sequencing is basically this RiboSeq can actively so the RiboSeq is based on the principle that actively translating ribosomes protect specific regions of mRNA from ribonuclearization. These ribosome protected mRNA fragments are called ribosome footprints, and the number of ribosome footprints indicate the active level of translation. This can be used to identify actively translating ribosomes, which is useful for studying protein synthesis. And when these fragments are sequenced, ribosome portions on the mRNAs can be precisely mapped, and translation activity can be studied in detail. So here what you're trying to see is which of these mRNAs are actually translating into protein. You capture the mRNA in a form where the ribose is still bound to the mRNA. Ribosome is still bound to the mRNA, so that you can have an idea that yes, this particular transcript is going to be used for protein synthesis, right? So you can study the protein synthesis using RiboSeq for sequencing of ribosome attached mRNAs, right? So RiboSeq initially involves freezing the ribosomes and treating the cells with ribonuclease enzymes to digest unprotected mRNAs and isolate ribose footprints. These ribosome protected mRNA fragments are then converted into compatible, again, cDNA libraries and analyzed using some Illumina technology or some next generation technology, right? Yet the only difference is that you only want the part of the mRNA that is attached to the ribose, right? The ribosome attached mRNA is the important part. And so why we need this? Because traditional RNA sequencing cannot reveal which mRNAs are being translated into proteins. It only measures how much mRNA is present in a cell, and just because an mRNA is there does not mean it is being used to make a protein. Unlike traditional methods, riboseq focuses on only ribosome protected mRNAs that are being actively translated, right? So we started with ESTs, then we talked about microarrays, then we talked about bulk RNA sequencing, single cell RNA sequencing, and ribosequencing. So these kind of actually encompasses the whole transcriptomic landscape. There are more technologies present, but like these are the most widely used, and almost all information about RNAs and mRNAs in terms of what modifications there are, you can use direct RNA sequencing to find that. If you want to find just the counts, you do a bulk RNA sequencing. Just give me a minute. Hello, I'm পড়াচ্ছি। হ্যাঁ হ্যাঁ ঠিক আছে ঠিক আছে। Yeah, sorry for that. So you can find modifications on the RNA using direct RNA sequencing. If you just want to find the counts, how much of a particular gene is getting expressed or not, you can use RNA sequencing. If you want to find cell specific RNA behavior, mRNA expression, you can do single cell RNA sequencing. If you want to find downstream translation, if the particular mRNA is getting translated or not, you can do ribosequencing, right? So this kind of entails the whole transcriptomic landscape. Now we move on to proteomics, right? So similar to transcriptomics, proteomics is the study of the proteome, which is the complete set of proteins that are produced in the biological system. It provides direct insights into cellular functions because proteins carry out most of the gene functions in the cell. Proteomics focuses on the study of proteins, their structures, their interactions, functions. Proteomics can be used to study the expression of proteins. Additionally, it can reveal how proteins are modified, such as by post-translational modifications. It can also provide information on movement of proteins between different subcellular compartments, as well as their involvement in metabolic pathways and interaction with other proteins. So we have like kind of three different types of broadly proteomic principles, which is bottom-up proteomics. You chop it into smaller segments and then you do so for in proteomics we do not do we do sequencing through a technique called mass spectrometry. We'll touch upon it a little bit later, but yeah, so either you do bottom up, you have small peptides and you do peptide separation and then you see what these protein sequences are. Then you have larger peptides and then you also have like kind of a proteoform, which are like different folded proteins that are slightly different from each other, right? So you can have all these types of samples and then you can find what the amino sequences are, if there are modifications on the sequences somewhere, or where this particular amino acid or protein is getting generated, right? So you do digestion for protein digestion for the first two steps, because you have protein in a folded form, you have to unfold it and then fragment it. But in top down, you generally do not do in digestion. You just do separate the protein and try to find things about it. So mostly structure and so on. So this I have not put it in the PPT, but this is actually a very interesting blog I found. Yeah, so these talk about different protein purification methods. Mainly it is done so how proteins are purified, because you need to purify the protein before doing the sequencing, right? So either you purify it on the basis of solubility, size, charge, binding specificity, right? So you can do centrifugation, which is done on the basis of so sorry on the basis of size. In terms of dialysis also you can do it, which is on the basis of charge as well. So that you can do column chromatography, which can differentiate between size, charge, and affinity to bind to another protein. You can do gel filtration. I think a lot of people do gel filtration in different methods. You can do ion exchange, so you can do cation and ion exchange chromatography. So the idea is that different types of moieties that are used from weakly to strong, very strongly charged, thus allowing a huge range of molecules the ability to interact, how they interact, and so on. Affinity chromatography is basically it will bind to some other protein and you kind of purify it on the basis of that. Electrophoresis is the most common one. I think a lot of people have, a lot of you might have done some of these experiments in college or not school, probably college only, SDS page and so on. Gel electrophoresis. Uh, yeah, SDS page I was talking about, paper electrophoresis. So you can have a lot of these techniques, but you can have antibody purification, where you have a particular antibody made to bind to a particular protein, so that you have in a kind of a test tube, you have antibodies at the bottom. You put the whole solution. the 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 proteins that are complementary to this antibody will go and bind to the antibody, and you can wash the rest off and you can purify it from the antibodies itself. So that is also very very widely used. Then you have techniques like Western blotting, which also uses antibodies, and you have a signal in terms of a chemiluminescent signal, which after binding, you can detect if the binding has happened or not. And you have ELISA also, which is a most frequently used technique, and mass spectrometry. So basically, uh, uh, yeah, this is one of the most important analytical techniques available, and part of the power is of the technique lies in its ability to detect minute quantities of material, which is 100 picograms or less. A mass spectrometer determines the mass of a molecule by measuring the mass to charge ratio of its ion. Ions are generated by inducing either the loss or gain of a charge from neutral species. Once formed, ions are electrostatically directed to a mass analyzer, where they are separated according to the mass to charge ratio, and they are finally detected. In general, the results of a MSMS or MS experiment are compared to the standards in a database to determine the components of the original sample. So this is a blog I found very useful in terms of protein purification. So if you are interested to know more, you can probably go through this. Okay. Okay. Now we talk a little bit about mass spectrometry, because that is the most important type of technology for proteomics, and kind of the most high end till now. Okay. So yeah, as I was telling, mass spectrometry is an analytical tool useful for measuring the mass to size ratio of one or more molecules present in a sample. These measurements can often be used to calculate the exact molecular weight of the sample components as well. Typically, mass spectrometers can be used to identify unknown compounds via molecular weight determination, to quantify known compounds, and to determine structure and chemical properties of molecules. How does a mass spectrometer perform such a feat? Each mass spectrometer consists of at least three components, which is the ionization source, the mass analyzer, and the ion detection system. So it looks something like this, just to give you an idea. So this is it first it goes to the ion source, then to the mass analyzer, then to the detector, and finally you process the signals, right? So the ionization source, in this case, molecules are converted to gas phase ions so that they can be moved about and manipulated by external electric fields and magnetic fields. You use something called nano electrospray ionization, which is somewhat similar to how cars are industrially painted. This method allows for creation of positively or negatively charged ions depending on experimental requirements. Nanoelectrospray ionization can directly couple the outlet of a small scale chromatography column directly into the inlet of a mass spectrometer. So you have the chromatography column where you are separating the proteins, and then from that you have some dispenser which can convert it into some gas phase ion and directly be inserted into the mass spectrometer. So then you have the mass analyzer. One ionized, the ions are sorted and then separated according to the mass to charge ratios. There are a number of mass analyzers currently available, each of which has trade-offs related to speed of operation, resolution of separation, and other optical requirements. So some of these are written here. So which quadrupole is one of them, ion trap is another, TOF time of flight is again another one. So these are all types of mass analyzers present. So this in Broad Institute, I hope you have heard about Broad Institute. This blog is taken from there. I'll just give you the source. Broad Institute is basically the nomad this is the blog from Broad Institute. You can take a look here. This content is taken from there only. You can take a look into other of their blogs. They generally have very detailed blogs about these kind of things. Okay. Coming back, yeah, you have a mass analyzer. It works in concert with the ion detection system again. Now, what is the ion detection system? The separated ions that are measured, that are then measured and sent to a data system where these m by z mass to charge ratios are stored along with their relative abundance. A mass spectrum is simply the m by z ratio. So this is what a mass spectrum look like. uh, it is an m by z ratio of the ions present in a sample plotted against their intensities. Each peak in a mass spectrum shows component of unique mass to charge ratio in a sample, and the heights of the peak tell you about the relative abundance of the various components in that sample, right? So this is how basically a mass spectrum would look like. And again, this was the image that I was showing you. So you have these three are the main important components. You have an ionization source, you have a mass analyzer which sorts the ions based on mass to charge ratios, and you have a detector, and then you have a signal processor, right? So this is an overview of how a mass spectrometer looks like, right? So these kind of things, quadrupole, ion trap, and time of flight, these kind of things you'll hear a lot. So these are different technologies by which you can quantify this m by z ratios and kind of sort this, and yeah, so this is the working of a mass spectrometer, right? So if you want to do like so next part, this is the I think the last part, yeah. So this part we are going to talk about label free and with label methods for these MSMS techniques, mass spectrometric techniques. So label based approaches includes the incorporation of the stable isotope label at different stages depending on the type of technique deployed, and simultaneously profiling of more than one biological sample in a single MS run. The label samples are mixed in an equivalent ratio and are analyzed by LC-MSMS. The quantification is based on the comparison of peak intensity ratio of the labeled peptide pairs. In case of label-free methods, the samples are separately prepared and are subjected to individual analysis. So if you want to do multiple samples together, you have to kind of label it accordingly. So say for example, you have two conditions. You are giving one light tag to one condition and then a heavy tag, and then you mix and then you do the analysis, right? In case of label free methods, you have to do it separately, right? And it becomes a little bit difficult when you are comparing like the results of label free methods. Your other conditions should be exactly same, because there can be variability in the experiments, and if Your results can be artifacts to these variability, so you need to be very careful about batch effects and so on in terms of label free methods. So these are some labeling techniques. I'll this I think I have this paper somewhere downloaded. One second. I think this is the paper. Yeah. So if you, I don't think this is available freely, so that is why if you want to, I don't think that Google Meet actually supports PDF, does it? No, it does not support sending PDFs, but yeah, I'll share the name of the paper here. I don't think this is available freely, but if you can get a hold of this, please take a look. This is a book chapter, not a paper. Yeah. So, oh no, this is for this particular plot. Oh, okay, okay. Oh, is it? Oh, no, this paper. I'll just give me a second. I think I can bring this up. I think this is available. Just give me a second. Huh, got it. Yeah, this is this paper. So they talk a lot about how to do quantitative proteomics, and yeah, this table is basically this one. So different types of labeling technology. So for example, Silac, right? Let's go back to the PPT. I'll put the link in the description. Yeah, let's go back to the PPT. Yeah, so you have different types of labeling methods. So for example, you have something called Silac. It incorporates at organism level, and but it is not applicable to human samples. Arginine to proline conversion is what it does, so it is expensive and slow. Enzymes other than trypsin and lysine may produce non-quantifiable peptides. Auxotropes for the labeled amino acid. So there are the labeling technology techniques are basically some are applicable to humans, some are not. So these are basically metabolic labeling. So you're changing the amino acid. In some cases, these are chemical labelings, which are basically only incorporates at protein level, but in this case this incorporates at organism level and so on. So you can do a lot of these. There are a lot of these labeling technologies that you can use to label different conditions based on your experimental procedure. Yeah, so yeah, again, so you have label free quantification when you do where you do the MS analysis separately, and if you have you want to label, you do metabolic labeling of, for example, Silac or ICAT for your chemical labeling, and then you do the MS analysis. So yeah, so that's all in terms of the content of this week's. We will move into the assignment questions. If you have any questions, please ask, or else we'll move on to the assignment problems. Okay, let's move on then. Yeah, sorry. Okay, so today's questions are mostly theoretical. You do not need to work out anything. So yeah, let's go on. So one thing I want to say is that if you see these square boxes, these are basically, this means that they multiple options correct, and if you see round circles, that means it's a single option correct. Okay, so okay, so let's get started with the yeah. Please write the answers in the chat box. We'll discuss. Oh, I thought. Sorry, let me check. In week 5, the option is given for only one, no multiple option for this particular question. Okay, but in my what was shown to me, this is a multiple option correct. So that is also my understanding, that if you have the square boxes, these are multiple option correct, and if you have the single like circles, that means you have like single option correct. That was my understanding, and in this case also, like, it is multiple option correct. So I'm not sure about the this thing. So if it was like only one option correct and no multiple option for this particular question, I think you need to take this up with the NPTEL team. I understood. Yeah, you need to like write a mail to the technical team. Probably they'll they'll help you out with this. Or you can bring this up in the discussion forum or something like that. Yeah. Maybe writing to the NPTEL team would be good. Yeah. Okay. Yeah. So epigenomics is basically modifications on the DNA. So that can be DNA methylation, that can be histone modifications, that can be chromatin organizations. So, and syntenic transcription is not part of this. We are only talking about DNA in terms of epigenomics. So these three options are correct. Okay, moving on. an element. Oh, okay. Yeah, basically all of these. Here they have given all the answers, but yeah, all of these is the correct answer. So from the function, structure, and sequence, all of these can be due to conservation, due to evolutionary forces, and there will be slight differences in all of these things across different species, even if the species are very similar. Say, for example, chimps and humans, you'll have slight difference in function of a particular protein, slight difference in structure or sequence, anything. Sequence is the main part. Based on the sequence, you have difference in structure or function, right? So okay. Yeah, that's correct. Express sequence tags. Yeah. The answer is post translation modification is something. I think someone answered D. If I'm not correct. No, 1 and 3. Oh, okay. D was the. Yeah, so protein transport you cannot. For protein transport, the cell has to be live, and you have to track. So for protein transport, what you need to do is put a tagged protein inside a cell, and then you see how the protein is getting transported from different parts of the cell, so from nucleus to cytoplasm or somewhere to somewhere. So protein transport is a very difficult thing to study, right? But protein degradation you can study. You can do it in a simulated environment. And also post-translational modification of the protein also you can study, but protein transport is very hard to study. Enzyme kinetics also you can study, but not through proteomics. That's a different field altogether, right? So yeah, moving on. Wait a minute again. Okay. Uh, the answers are basically ion exchange, affinity reagents, and peptide IDs all. HILIC is a technique. So that's a liquid chromatography technique to do enrichment, but HILIP does not exist, I think. Yeah, ion exchange, affinity resins, peptide IDs, all like ion exchange. You can, all these are types of chromatography techniques where you can use to, which you can use to enrich the peptide contents, right? So, yeah, and HILIC is something. So I'll just quickly show you. Yeah, so this is hydrophilic interaction chromatography. This you can use to do enrichment of peptides. HILIP does not exist. Yeah, okay. Moving on. think this is a single option correct. You have circles, so this is a single option correct, and the answer is DNA and protein. So ChIP-seq is basically chromatin precipitated. So you have an antibody or some protein that goes and binds into a particular protein that is bound to the DNA, right? So you have a DNA sequence, then you have a protein on top of the DNA sequence. What you're doing is you're taking another antibody, and then the antibody will go and bind to the protein that is on top of the DNA sequence, and it will precipitate. Now you have the protein DNA complex, and then you can only sequence the DNA that is attached to that particular protein. So you can tell the motif of the DNA at which the protein comes and binds. So that is the idea of ChIP-seq, right? So it kind of tests the interaction between DNA and proteins. So, and yeah, this is a single option correct. So you have, when you have singles, you have circles like this, these are single option correct. When you have boxes like these are multi option correct, right? So yeah. Okay, the correct answer is the first one. So first you ligate the reverse transcript split, then you do the reverse transcription. So the split is basically the primer for the reverse transcriptase. Then you do reverse transcription, then you do the sequencing of the ligation of the sequencing adapters, and then you sequence the RNA strand. So first you do the reverse transcription, right? So for that you need to have the RT split, and then you do the reverse transcription, then you do the rest of the thing. So the first option is correct. Okay. If you have any doubts, please ask. But yeah, this it's kind of intuitive. You first do the reverse transcription, then you do the adapter ligation and the sequencing. Four. Okay, again, uh, why not chip SS? As I said, the chip is kind of it's the idea is that you have a antibody that is specific to a protein bound to the DNA. Then you pull down the DNA along with the protein using the antibody, and then you can check what proteins are there and what is the sequence of the DNA and so on. So you can also detect epigenetic modifications with chip assays as well. So chromatin immunoprecipitation, chip stands for chromatin immunoprecipitation. So like all of the four options are DNA methylation analysis you can do using, say for example, nanopore technology helps you to get the data of the modified bases as well. DNA protein interaction analysis you can do, which is kind of chip assays in general. So chip and DNA protein interaction analysis kind of the similar option, right? And chromatin accessibility analysis also you can do. So all of the options are correct. Cheap assays is also correct, right? So yeah, let's move on. Okay, I think so. Single cell RNA sequencing, I think you gave all of the above. So single cell RNA sequencing cannot tell you anything about translation, right? It will only tell you about. It's basically normal RNA sequencing, but done at single cell resolution, right? each cell you can extract and sequence the transcripts, right? But it does not tell you anything about the translation. The translation will be you can get to know about translation if you do riboseq, if the ribosome is attached or not attached, right? But you cannot know about translation from single cell RNA sequencing. So rest of the options are correct, but for translation information from transcriptome, you need to do riboseq, right? Okay, moving on. Okay. Okay. Yeah, you're correct. So, BRU-seq is basically bromoiodinine. You tag the sequence with that, and basically, a brick-seq is basically after tagging with bromoiodine, you sequence the rest of the molecule. So, yeah, 2 on 3 and correct. So, RiboSeq can tell you about translation. As I was telling you from the earlier question, RiboSeq will tell you uh, if a ribosome is bound to the mRNA, which in turn can tell you if it is uh, ready for translation or not. So for the translation information, you have to use a RiboSeq. So yeah, it can quantify RNA translation. Okay, if you are confused between determination and quantification, the idea is that you can very much quantify that, because you can be count the number of mRNAs that are attached to the ribosome. So you can quantify that these many mRNA transcripts had ribosomes attached and these were going for translation. So a certain percentage of these transcripts were going for translation. So you can basically quantify RNA transcription if you are confused between determination and quantification. It can certainly determine, but it can very specifically quantify, right? So that is clear. Okay, no answers, but yeah, this is quite simple. So yeah, biosome is not something that is there. Like, I don't think that exists, but yeah, monosome, polysome, polyribosome, everything is correct. Biosome is not a correct term also, I think. So yeah, moving on. Yeah, the answer is quad 6. So these are different types of single cell RNA technologies. We have not discussed this in this session, but I think Sir has discussed it a little bit. So yeah, the answer is quad 6. Yeah. Yeah. Yeah, it's last week. I'll not go into the details of these because I have not covered it here. But yeah, these are different types of sequencing, single cell RNA sequencing methods. If you want to give it a read, yeah, I think Sir has also covered in quite detail. So yeah, yeah, it's all. I think I took the name of a couple of them while discussing mass analyzers, but yeah, these are different types of techniques by which the mass to charge ratio of the ions are sorted inside the mass analyzer. So yeah, we have not gotten into very details of these, but yeah, these are different types for this. No, the answer is it'll be either isotope labeled protein because it's done together, right? So when you are using p-silac or any other type of tagging technique, so you're taking all of the proteins together and doing the mass spec. So either isotope labeled protein will be in the extremes of the time gradient, right? So because you're taking both of them together, right? So either can be in either end of the spectrum. So heavy and light and heavy, both these combinations can be possible. So since you're taking it together, it's either. Right. I hope I was clear. Yeah, so basically the idea is that you're taking both the proteins together, tagging them, and then doing the mass spectrometry, right? So across the time gradient, it can be heavy isotope at the beginning, light isotope at the end, or it can be reverse as well, light at the beginning and heavy at the end of either of the proteins. So it can be any combination possible. So it can be either. It can be either, right? So either isotope level protein can be present in across the two extremes of the time gradient. Yeah. Excuse me. No. So you're talking about this question, right? It's a time gradient, right? It's kind of initial and final, but according, like, it's not initial and final of the cell state. It is like initial and final of the time period of the experiment. So it is like it's a time gradient. So firstly what has been sorted and lastly what has been sorted. So it is kind of initial and final, but not in terms of like first this protein has been done, next that protein has been done, like that. Like not in terms of initially this particular m by z has been tagged, later that has been tagged. So not in that case. Like it's basically a time in terms of initial and final with respect to the experiment, not with respect to the cell types or cell specificity, right? Or sorry, proteins in the cell. Yeah, so it this answer is basically through the linker region. So basically the heavy tag will have deuterium or C13, and the light tag will have hydrogen atom in the linker region. So that is the important part. The answer is Silac. In Silac, you can do in vivo labeling. Less of them, you have to do the labeling first and then transfer. So, yeah. Yeah, the answer is both metabolic labeling and stage mass spec. We have not discussed about single stage and double stage in details, but yeah, so the idea is that you can do the Silac labeling, you can use Silac for metabolic labeling, and since you're using Silac for labeling, you can incorporate both your samples in one go and do the mass spec in one go. So you can do single stage mass spec, right? So for double stage, you need not do any labeling. Right? So, and finally, yeah, that is correct. The mass repeater and the mass normalizer. We have not discussed TMT today, but sir has discussed. So yeah, I think that is all from my side. So if you have any questions, you can ask, or we'll close the session for today. Sorry. Uh, yeah. Okay then. Thank you for joining. We'll see you next week, same time. Yeah. Thank you.